Hi everyone. So today we're going to learn about the secondary structure of protein. So we already know the primary structure of protein, which involve the peptide bond formation. Now we're going to learn about secondary structure of protein, and in secondary structure of protein we have two major conformation. One is helix and then another one is beta sheet, right? And then in helix, as you can see over here, that we have different types of helix present over here. We have an alpha helix, beta, we have gamma, and then we have pi helix. And then I'm going to tell you how I have written this 3.6, 310. I'm going to talk about this as well, so don't worry about it. And then in the beta sheet, we have parallel and anti-parallel. We're not going to deal about beta sheets in this video. We're going to talk about specifically about helix. So let us jump to the first one, that is alpha helix. So I'm going to tell you what is an alpha helix. So in let us just so as you can see over here, how many amino acids have I made? If you are able to spot, this is the first amino acid, right? This is the second one, this is the third one, this is the fourth one, and this is the fifth one, right? And then how am I going to number them? The first one I'm going to name it as n, the second one I'm going to name it as n plus 1, third one n plus 2, fourth one n plus 3, and fifth one n plus 4. In place of n, it is also possible that you write I, and then you could write I plus 1, and then I plus 2, and then so on. So that could also be done. So as you can see over here, that I have drawn 5 amino acid, and I have made it clear that they are joined with one another because they have a peptide bond, right? This is the peptide bond, right? This is the peptide bond, right? This is the peptide bond. Sorry about it. This is the peptide bond, right? So as you can see that they are joined by one another by peptide bond. Now this is actually a primary structure, but then in secondary structure what happens is there is formation of hydrogen bond as well. So when a hydrogen bond is formed between two atoms, what happens is the structure from a linear structure now starts to form a helical structure. So I'm just going to write it down. So in primary structure, what was the main force, what was the main bond which was involved? It was the peptide bond, right? In secondary structure, we have the, which bond do we have? We have the peptide bond that we have, obviously, but then we also have hydrogen bond involved. Okay, so I hope you're able to understand this. Now let us take this particular amino acid in the form of a helix. So I've taken this amino acid in the form of a helical structure. So this is actually a clockwise helix, right? Because it is going in the clockwise direction, right? Now the clockwise direction means it is left-handed. If it is right-handed, then it's going to go anti-clockwise. It's just going to go opposite. Now, as I told you that the first amino acid was named as I or N, and the second one continuously like N plus 1, N plus 2, or I plus 1, I plus 2. So I've just written out here. I'm just trying to show you like in a general form. So this is the axis, and then around this axis, we have the helical structure, and then this is the C double bond O group of the first amino acid. So I'm going to name it N, of course. And then this is the second amino acid, right? So I'm going to name it, am I going to name it N2 or am I going to name it N plus 1? I'm going to name N plus 1, right? So this is N plus 1. What about this one? This is the third one. This is going to go N plus 2. What about this one? this is going to do n plus 3, right? And then the last one, this one, this will be n plus 4. Okay, so now as you can see in this helical structure, a hydrogen is attached to a nitrogen, which is an electronegative compound. And then as you know that an electronegative compound, when it is when a hydrogen is attached to an electronegative compound, there is possibility that it can form bond with another electronegative compound. So out here, as you can see that when nitrogen, let me write it down here, since nitrogen is forming a hydrogen bond, it is possible that any of the other electronegative compound can form bonds with the hydrogen. So there is a possibility, right? And out here I see a possibility that the hydrogen can form a bond with oxygen, right? Yes, there is a possibility. So let me just draw a hydrogen bond over here. Okay, so now we have a hydrogen bond. So similarly, what happens is the second okay, so yes, so as you can see over here that I have shown you how does a hydrogen bond forms. So let us learn about other stuffs as well. So this is a helical structure, right? This is a helical structure. And then the helical structure, now this is formed between n and then n plus 4. That's why it is known as an alpha helix. Okay, now in this particular alpha helix, this is actually one turn, right? One turn, that means this is one pitch. And then in one turn there are almost 3.66 amino acids present. That means in particular one turn. So as you know that this is one groove and then another groove, and then over here we have around 3.6 amino acid which is taking part, right? And then the pitch is actually the height of this this structure is 5.4 angstrom. And then there is delta z. Delta z means like what is the distance between two amino acid, like what is the vertical distance between two amino acid. For example, let me just show you. This is one amino acid, right? And this is another amino acid. This is n plus 3, n plus 4. Now what is the distance between these two amino acid? That is actually the z, and it is also known as axial height or rise per residue, amino acid rise per residue. That means how much one amino acid is rising above the other. What is the vertical distance between these two amino acid? If I take this and this, and if I take this and this, if I take this and this. I hope you're able to understand. Okay, and then there is a formula which says like number of amino acid is equals to pitch divided by axial height. So you can always like interchange the formula, and then you can always, if they have given you pitch and the number of amino acid, then you can always interchange these formulas, and then you can take out the axial height as well, right? Now okay, now let us see another one, that is how much degree, or how much degree of rotation or degree is formed between one amino acid and another amino acid. So as you can see over here, let me take another one, let's take green. So as you can see over here that this is one amino acid, right? This is one amino acid, and this is actually the angle that is forming, and this is actually the particular angle that it is forming. So this is 90 degree, right? So this will be 100 degrees. So an angle between two amino acid with respect to the axial axial height, with respect to the axis, center axis is actually 100 degrees. So I'm just going to write it down over here. It's actually 100 degrees. Okay. So now I hope you're able to understand that how does a hydrogen bond form between two like between like different amino acids, and this is the reason why they form this helical structure, because before they're not forming any hydrogen bond. Afterwards, what will happen is they will form hydrogen bond, and then they'll undergo a structural conformation, that is helical structure. Now I've shown you in a helical form. Now let us just try to see in a linear form how does it look like. So out here, this is actually the amino acids which will be involved in the formation of hydrogen bonds. So out here I have taken certain things. Let us see. So as I said that in the first carbonyl of the first amino acid will be involved, and then the NH group of the fifth amino acid will be involved, which will be able to form the alpha helix, right? So if I just want to show you, this is the first carbonyl, right? This is the first amino acid carbonyl group, and this is the amino group of the fifth carbonyl, right? So this hydrogen will try to form a bond with this oxygen, right? So this will be termed as alpha helix. That means whenever a carbonyl group of the first amino acid will try to form bond with the fifth amino acid of the with the amino group of the fifth amino acid, then it will be termed as alpha helix. Okay, so I hope now you're able to understand how does it look like in a linear form as well, right? Now I was talking about 3.613. If you remember, I told you something about the 3.613 helix. Now the reason the 3.6 is actually the number of amino acid which is present per pitch, right? That means amino acid which is present per turn. Now what is this 13? Now let us see what exactly does this 13 means. So now the 13 is actually the number of atoms which is involved in the formation of hydrogen bonds. Now let us count from this side to this side, right? So let us first count the backbone. So from here it is there. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, right? So from the backbone there is 11 atoms involved. Now what about the other two atoms? That is 11. We have 11 over here, and then this is also involved, right? This is the main one that is involved in the hydrogen bond. So 12 and then 13. So there are 13 atoms which are involved in the formation of the alpha helix. Therefore the name itself says 3.6 and then 13. That means 3.6 is actually the number of amino acid present in the pitch, and 13 is the number of atoms which is responsible for the formation of hydrogen bond. Now if I just show you another one, that is, let us take another one. Okay, so I've given some details about what exactly is the pitch, the number of residue per turn in different helixes. So if you see over here, this is the alpha helix, and there are number of residue, that is number of amino acid is 3.6. Then the pitch is 5.4 angstrom, and then the rise per residue, that is delta Z, is 1.5 angstrom. That means, what is a delta Z? That delta Z is actually the the vertical distance between two amino acids. Now if you see over here, we also have beta helix. Now in beta helix, which are the amino acids which will be responsible. So in the beta helix, as you can see over here, I'll just try to show you. So this was the alpha helix, right? I'm gonna rub this. I'm gonna show you the beta helix. So this was the alpha helix, right? So now beta helix is actually this one. So alpha helix, in alpha helix, this was involved and this was involved, right? From here also we had the C double bond O, and from here we had NH group. Now in beta, this will be involved and then this will be involved. Now from this we have the carbonyl group, but then from this we have the NH group, right? So I hope you're able to follow up. So I'm just gonna show you. So this hydrogen will form bond with this oxygen, and this will be termed as beta. Similarly, if this nitrogen forms, let me take another color. If this nitrogen forms bond with this oxygen, then it will be termed as gamma, right? And then if this nitrogen will form bond with this oxygen, then it will be termed as so as you can see over here, that this is now there are another possibility, that is last one, that is N and N plus 1. So this can form bond with this one, right? And then when this can form bond with this one, only 4 atoms will be involved in the formation of hydrogen bonds, and this one is known as the delta helix. So this was beta, this was gamma, and then this is the delta helix, right? Delta helix, I think I've not given the name, so let me just write down over here 0.6 and then 4. Why 0.6? Because 0.6 atoms are involved in the formation of this helix, and then 0.6 atoms are found in one turn, and then 4 atoms are involved in the formation of hydrogen bond, that is this one and this one, right? Therefore 0.6, and then we have subscript, that is 4. Now if you see in this diagram, you'll be able to see that there is alpha helix, beta helix, gamma helix. You can always count the number of atoms, and then you can just find out if it's the perfect one. We talked about 2.27, right? So let us just count 7. So where is the gamma? Okay, so 7. So let us see, 1, 2, 3, 4, 5, and then 6, and then 7, right? So 7. So now I hope you're able to understand what exactly do I mean. And then we have number of residue per turn, which is this one, and then we have a pi helix as well. So pi helix is I'm gonna write the number for the pi helix, which are the amino acids involved. So it is the first and the 6th amino acid. Okay, so 6th amino acid is responsible, and then we have rise per residue. Rise per residue means how much vertical distance is between 2 amino acids, and then it's also known as axial height, right? And then we have radius of helix. Radius, that means if this is a helix, Then what is the radius of the helix? So this is the radius of the helix, and then this is the pitch. That means one turn. So one turn, right? And then how many number of amino acid is present in per turn or per pitch. There is also one line over here: the more number of amino acid increases, distance decreases. That means, as you can see in the pi helix, the number of amino acid is increasing, and then the pitch is also decreasing, as you can see over here, right? Over here also, the amino acid, as it is increasing in number, then the distance between them is decreasing. Last common thing is that, what is the most common form of like helix? We learned about alpha, beta, gamma, delta, and then we also learned about pi. The most common form is alpha helix. The reason why it's alpha helix is because alpha helix forms a hydrogen bond which is like 180 degree. So as you can see over here, that it forms a 180 degree hydrogen bond, and 180 degree hydrogen bond is the most strongest form of hydrogen bond. Therefore, alpha helix is the most commonly found in the nature. Now let us see another one. Right-handed. It should be right-handed. That means the one that I drew up, it was left-handed, but right-handed means like it should be anti-clockwise, right? And the atoms are the first amino acid and the fifth amino acid. Now the amino acids which favors alpha helix are these 5 amino acids. Others also favor, but these are like the most favorable ones. That is methionine, alanine, leucine, glutamate, and then we have lysine. So these are the amino acids which favors alpha helix. So I hope you're able to understand today's video. As you can see that we talked about we talked about helix, and then about helix we learned about these also delta, I forgot to mention, and then also we learned about the axial height, the turn, the pitch, how does a hydrogen bond formation takes place, which are the amino acids which are involved in the formation of hydrogen bonds, and also like how many atoms are involved in hydrogen bond formation. And also I gave you a small detail about, you could learn this table. And then yes, thank you so much for joining this video. Please like, share and subscribe, and thank you so much.